Hey everybody, Mr. Murphy here. I'm going to take a look at chapter 4.3, which is all about eukaryotic cells. So eukaryotic cells are notable for the fact that they have organelles, and those organelles are basically just membrane enclosed segments inside of a cell. It's a cell within a cell in some regards. And each of those organelles is defined by the unique characteristics that they have that then yield specific functions. So they are sort of functional little units within the cell that massively increases the cell's efficiency and its ability to handle a dynamic environment. So when we look at these, we're going to go through each of the cells, each of the organelles, and here's a representation of an animal cell. You can see how it's a chaotic mix of all of these things. I've pointed this out before in class probably, but representations like this are great, for being able to visualize sort of the tangled mess of stuff, but it's also important for us to understand a few things like there's probably many mitochondria within the cell. There's many times of all of these things. The ER is actually this folded tube network, not even tube really, it's just this folds of membrane all throughout the three-dimensional cell. So when you're on a cellular level, all of these different organelles are made out of phospholipid membranes, just like everything else. And so, if you shrunk yourself down and were floating around inside of a cell, you really would have a hard time differentiating many of these things just purely based upon what the outside happens to be made out of. And so that's why we focus a lot on like the cross-sectional shape of a mitochondria, for instance, helps us to differentiate it between that and the ER or the Golgi, which does have a very, it's kind of a blob-like shape, but it's a very specific shape for what its function is, versus peroxisomes or lysosomes or these little circular dudes floating around. So plant cell, pretty much the same thing, except some notable differences. We have a huge vacuole. They have a cell wall on the outside of the cell, and chloroplasts as well, which photosynthesis, right? Ribosomes are probably the first thing to just address, and we mentioned this in the prokaryotic section. So they're in both types, and they're not membrane bound. So they are not really true organelles in the sense of having their own nuclear membrane. However, they are absolutely essential to everything that the cell does because it's the conversion unit for taking the information from DNA, RNA, and turning that into something that actually serves a purpose chemically and otherwise inside of the cell, which is proteins. So that's ribosomes. Like I said, they are not membrane bound. Okay, so they are different than many of the other organelles that we're about to discuss. In eukaryotes, they can float around in the cytoplasm, and then they can also land in the endoplasmic reticulum. Interestingly, they're also found inside of the mitochondria and the chloroplast. And even more interesting, the ribosomes that are found in the mitochondria and the chloroplast belong to the mitochondria and the chloroplast and serve to produce proteins made by the DNA of the mitochondria or the DNA of the chloroplast, which, think back to chapter 1, we introduced the term endosymbiotic theory as we look at where did the mitochondria and the chloroplast even come from. They were once their own cells. The nucleus, typically the largest organelle, it's one that is visible in most cells, eukaryotic cells that we look at under a light microscope. It's the location of DNA and DNA replication. It houses it, it protects it. It contains the nucleolus, which is where ribosomes are assembled to begin with, because ribosomes are actually made out of RNA and various associated proteins. It is surrounded by the nuclear envelope, which is another double or phospholipid bilayer that is there specifically to protect that DNA, as the DNA is the sort of instructional basis for everything that's going on inside of the cell. It needs to be kept safe, and so the nucleus is all about keeping it safe, but also keeping that information accessible to the ribosomes that are going to need to acquire that information. Just to clarify, so we understand, the information of DNA is converted into RNA and then leaves the nucleus. So ribosomes aren't like coming into the nucleus to get things. The information is sent out. Inside the nucleus, typically we find DNA coiled up into chromatin, and then it is condensed into these even tighter packages called chromosomes, specifically during mitosis and meiosis, the splitting of the nucleus. The endomembrane system sort of incorporates many of the remaining organelles and refers to basically the system of transport for the various proteins that are produced inside of the cell so that they can end up at their intended location, whether that is within the cytoplasm, within one of these organelles, or outside of the cell. Here's a representation of this, and we'll look at this diagram a lot in class. We have the nucleus sort of centrally located generally in the cell. The endoplasmic reticulum is surrounding the nucleus, and really it's sort of intermixed with all of this because, like I said, the ER really fills the cell. Adjacent to the endoplasmic reticulum, though, you will find the Golgi, which is a series of these sort of flattened stacks or little blobs here. And budding off of these are different vesicles. A vesicle is just a small little phospholipid orb that is carrying different products inside of the cell for a whole host of reasons. And those can become lysosomes. Those can serve as export vesicles to remove things from the cell or deliver, for instance, like enzymes from your pancreas or whatever, getting ultimately delivered to the inside of your intestine. Those are going to need to leave those pancreatic cells. So the Golgi really is the sort of packaging network of the cell, and based upon the different chemical cues that are going on with the various proteins and associated molecules, it's going to indicate what direction things need to continue. The ER, like I said, interconnected membranes. We have rough ER, which is rough, and the smooth ER, which is smooth. The rough ER, we call it rough because it has all these little dots peppered throughout it, and that's the ribosomes. And those are ribosomes that are delivering proteins that need to be transported through the ER. So the endoplasmic reticulum has kind of two regions, the rough ER, which tends to be more nucleus adjacent because that's where the ribosomes are getting their information and then delivering their proteins to the ER. And then there's the smooth ER, which still serves as basically a modification center for the different materials that are within the ER. And it is also associated much more with lipids and synthesis of other molecules than proteins inside of the cell in packaging and delivering and all of that. The Golgi then, like I said, is those little flattened sacs, and that is picking up these products of the ER and continuing them on to where they need to go. And all of that is chemically mediated. It's all just based upon the attraction of different molecules and helper proteins and things that are going to be attracted to specific receptor sites and things like that on the vesicles that are coming from the ER, and the Golgi then is basically just a big chemistry set that's manipulating things and changing things and preparing them or finalizing them for what they need to be in order to serve their function. So the Golgi does have three different regions, and we'll talk more about that in class in terms of like the scope of this that does matter. But we have the cis region, which is adjacent to the ER, the trans region in the middle, and then we have the medial region, which I'm sorry, I said that backwards. This is not in the order I wanted it. With the cis region that's next to the ER, the trans region, which is facing the cell membrane, and then the medial region is in the middle, and that's a transition phase between the newly received proteins from the ER. Stuff needs to happen sort of in the medial region, and then the trans region is where these proteins are being packed up into vesicles or whatever to be sent off to, again, wherever they happen to be going. Primary lysosomes originate from the Golgi. So when we talk about how all of these different organelles are made out of the same types of membranes, it kind of comes together and makes sense why a little piece of the phospholipid membrane, and I gotta go back because I don't know if I said that right, that all the different organelles are made out of the same phospholipid membranes, not the same proteins. But as pieces of that Golgi come off and bud off into their own little vesicles, those vesicles there may contain the ingredients to sort of be a lysosome, that the digestive enzymes that were produced by the ribosome, sent to the ER, delivered to the Golgi, packaged together and allowed to form this little bubble of phospholipids. That's what a lysosome is. It's a piece right off of the Golgi. And, if you're thinking about like, well, if the Golgi keeps losing pieces of itself, how's it getting new ones? Well, remember that the vesicles came from the ER. So the ER's membrane is constantly being transferred over to the Golgi, and then the Golgi membrane is constantly being popped off as these vesicles, and then those end up being sometimes delivered different places inside of the cell, or maybe even being delivered to the cell membrane. And the whole vice versa process happens as well. Cell membrane gets bubbled into vesicles as the cell brings things in, and it's all this cycling. And then there are, in the smooth ER and elsewhere, mechanisms to continuously produce new membrane material as well. But it's a pretty crazy cycle and exchange. And that's what defines the endomembrane system, is that all of these things really are functioning together for this ultimate goal of producing things and getting them to where they need to be. So when molecules are taken in by the cell through phagocytosis, which is the cell eating, that's the taking material from the outside of the cell and pushing in a piece of the membrane, making a little vesicle and bringing that in, that can go a variety of places. And one of the places is that they fuse with these primary lysosomes and become what are called secondary lysosomes. And this is not a level that's particularly important for us to understand, but we can see from simple diagrams like this what's going on here, that the primary lysosome's been produced by the Golgi, new stuff from outside of the cell's brought in and mixed, combined with that primary lysosome, and that's where the lysosome now can do its digestive wonders to annihilate whatever that was brought into the cell. So maybe that annihilation is happening in order to liberate monomers of lipids or carbs or proteins for the cell itself to use. Maybe this is a cell that's taking in, potentially bad or foreign or whatever stuff that's not supposed to be in the cell or around the cell or in the body or whatever, and annihilating it. Any number of things. But the lysosome is kind of this Pac-Man within the cell, and one of its jobs is to gobble up and eat the stuff that's the little energy pellets that are coming into the cell that are being absorbed. The lysosome can annihilate those. Not to mention that it can do a whole bunch of other things in terms of cleaning up the cell itself and maintaining homeostasis in that way. So here you can see a electron microscope view of a cell, and it's a little more chaotic and bumpy than we sometimes like to pretend cells are. And here you can see this is something coming in through phagocytosis. Here's the phagosome, which is taken into the cell, and then it's going to join up with the primary lysosome, and when that happens, it takes on a different appearance as those enzymes are breaking down the materials of that. So here you can see sort of the breakdown and the autophagy here is the programmed destruction of cell components that the lysosome is also responsible for. So the cell's constantly making new stuff, but our cells aren't constantly just getting bigger and bigger and bigger, and so there's this sort of recycling loop that's happening. And thankfully we have had billions of years to become really efficient at it, but the process does appear wasteful until we understand more about why it works, that the cell's constantly making new things and simultaneously breaking down things, and then makes new things and breaks them down, and it keeps the cycle forever going. There are different storage diseases associated with lysosomes failing, and those are some things which we might discuss in class. So the mitochondria, I hopefully don't have to say too much about. It's the powerhouse of the cell, right? Make sure we understand that this is where chemical energy that's available to the cell is primarily produced, and that is taking things like glucose, or specifically glucose, and breaking that down and using the energy of that bond to charge up an alternate energy molecule like ATP that's a little bit more universally available and accepted by a variety of proteins and organelles within the cell. And an important point right here at the bottom, cells that require a lot of energy often have more mitochondria. So don't get it trapped in your head that it's like the mitochondria. No, first off, it doesn't even make sense because mitochondria is a plural. It's mitochondrion if it was singular. But cells can be jam-packed full of mitochondria. If it's a muscle cell, a major muscle cell in your body, or a major complex of muscle cells, there's going to have tons of mitochondria in order to facilitate all of the energy that's needed to make that work. Because remember, we talked about how small cells are, and yet, when we flail our arms around and stuff and move our legs, that's a really large structure compared to the size of an individual cell. And so in order to make that happen, those cells are going to need a ton of energy all at once. So the mitochondria has its own membrane, just like pretty much everything else inside of the cell. It actually has two membranes, so it itself has increased efficiency in its ability to move things across those membranes, and we'll dig much more deeply into that when we talk about cell energy and cellular respiration. So here's the mitochondrion. Cool. Wrapping up a few more things here. So plastids, which are found in plants and algae, and they can differentiate into different types of organelles, some of which, like it says, can be used for storage. But the primary star plastid that we discuss in this class is chloroplasts, which contain chlorophyll, which they use to capture solar energy, and that's where glucose is ultimately produced for living organisms. Here you can see the chloroplast itself. Another one that, like the mitochondria, is comparatively more complicated than most of the other organelles, which again kind of reaches back to its endosymbiotic origin, as it was once its own independent living thing, but now has become co-opted and associated with plant cells. There are a couple more. These are not ones that we're going to specifically quiz you on or anything like that, but still important to or valuable to know. Peroxisomes, that's where we have the breakdown of things like H2O2, peroxide, right? Peroxidase enzyme, which we've talked about. And then glyoxysomes. These are found in plants. They're used for converting lipids to carbohydrates. And so there's different evolutionary selective pressures that have led certain types of plants to have more of these that might perhaps be starved of the ingredients that they need to produce a sufficient number of carbohydrates, or the environment that they live in necessitates that they have this mechanism. So that's not one that we'll see much. Peroxisomes will pop up on diagrams sometimes. I think I've addressed the chloroplast pretty well. We're going to get into so much more with that when we get to the energy unit. So inside of the chloroplast, there are different components like the granum, which is a stack of these thylakoids, which are the little pancakes inside of the chloroplast. And on that chloroplast picture, you can see tons and tons of these all associated together, these stacks of flattened discs. So each one of these is a stack of these little membranes across which the reactions of photosynthesis are actually happening. Random picture of a peroxisome, please enjoy. Vacuoles are found in both types, plant and animal, but or rather plant, animal, and fungi, but primarily in plants and fungi, as it serves multiple roles, not just storage of water, but also production of pressure, turgor pressure. So plant cells hold a lot of weight. Look at a tree, it's big, it's heavy, and those cells have a lot of force on them. Or imagine like a flower, the stem of a flower, and maybe you forget to water it and it starts to wilt, right? It's wilting because it's losing the pressure in its cells. Its cells are becoming squishy, and so its weight is allowing it to collapse a little bit. And then you remember to water it, and next thing you know, the next day it's up straight because it's filled those vacuoles back up. Cool. Vacuoles can be used in different ways. Like reproduction sounds weird, but basically it just means that certain flowers can have different pigments inside of them that facilitates then attracting certain pollinators and things like that. So there's a whole variety of functions to the vacuole, but primary thing to know is it stores a lot of water and it provides that pressure for plants. Contractile vacuoles, just to bring in the forgotten other eukaryote, protists utilize contractile vacuoles as well. Protists are a separate kingdom of life, right? Tiny little organisms. You find them in pond water and things. They're everywhere, but that's a common place where you could scoop up a droplet of pond water and look at it under a microscope. You'll see a ton of really cool things that'll also make you feel really not okay with swimming in a pond. But they have contractile vacuoles where they can actually specifically like shoot out water in order to balance their own salts and solutes inside of them, which is cool, but also produces a whole lot of force. So they can actually produce some movement through that as well. And that is the end of our eukaryotic section. So a lot of different cellular components, and it's important to really have a good understanding of the basic function of each of these organelles. So that way in this class, as we dig into deeper concepts, hopefully that becomes a little bit more palatable because we're not so worried about like, wait, what does the Golgi do? And stuff like that. So if you have questions, let me know. And the next section is about the cytoskeleton.